Welcome back to the Neuroblastoma Parent Symposium 2023. My name is Nick Bird and I am moderating this next session on advocacy in research. This session is going to be brought to you by Donna Ludwinski, Director of Research Advocacy at Solving Kids Cancer in New York and Family Support Advocate at Solving Kids Cancer UK, together with Emily Hall, Research Engagement and Advocacy Manager at Solving Kids Cancer UK. Then we're going to be hearing from Dr. Wendy London, Director of Clinical Translational Investigation Programme at Dana-Farber Cancer Institute, and Professor Maja Beck-Popovic, Head of the Pediatric Hematology and Oncology Unit at the University of Lausanne in Switzerland. Just to remind you again, the chat function on the right-hand side of the screen is where you can submit your questions, and we're going to have time after the speakers for a general discussion at the end of the session. And so with that, I would like to introduce you to Donna and Emily, who are going to present on what is research advocacy.
Great. Thank you, Nick. Get our slides up. Well, appreciate the opportunity to share today. A little bit of further introduction of why am I interested in neuroblastoma? Well, I had a child who was diagnosed at 6 years old in 1991, which was before there was an internet. And so obviously it was another era. He went through, even with a very poor prognosis, he went through chemo, surgery, radiation, and a bone marrow transplant before peripheral stem cells were used. They didn't even use MIBG scans. This was at Boston Children's. And surprisingly, he relapsed very, very late. It was 13 years later when he was in college. And then he went through almost 5 years of treatment in 5 different states. So that's where my background in neuroblastoma comes from. The year he died in 2010, I applied for an advocate scholarship to go to the ASCO meeting. That's the largest oncology meeting in, I think, the world. There's usually about 30,000 oncologists and researchers there. There were about 300 advocates that went to that meeting. And that's the first time I heard the word research advocacy. So if you want to slip to the next slide. The term research advocacy actually covers a range, covers a range of concepts and activities. And I met a relatively young woman there. She was a survivor of inflammatory breast cancer. And she said she was a research advocate. And she had applied to go to a boot camp to get trained as a research advocate in the breast cancer world. And now it's called Project Lead. And she said it was very competitive to get into this boot camp. And this is the part that impressed me. She had a PhD in biomedical engineering, and she had a hard time getting it. So this is the beginning of this fascination with this concept of research advocacy. It's not necessarily policy. It's not necessarily your traditional patient advocacy. It's getting involved a little bit more in the research enterprise and the drug development life cycle. So next slide. So we all start out as advocating for our children. And so, next. Oh, you are on I'm looking at the wrong screen, sorry. We start out advocating for our children based on their needs. And then further down the road, for those who have the aptitude and the interest, research advocacy gives you an opportunity to really get involved in trying to improve things systematically for the whole population of children, not just an individual child. So next slide. So research advocates are really involved in the patient community, understand the needs and what's happening there, but also are involved with the researchers, the investigators, clinicians. And so there's a way of bringing those two worlds together. So the role of a research advocate can actually be quite varied. You can get involved in influencing the direction of research and demonstrating the value of what the patient perspective is in the research, and then empowering patients and parents to make sure their voice is heard. And then you can also be working on specific research projects. It might be reviewing protocols or concepts. It could even be participating on study teams. And then in the next slide, you can see that there's actually quite a range of time points that advocates can get involved. This slide was actually made by Leona Knox when we were working on a chapter in a textbook on pediatric cancer drug development, what's the role of advocates? And so here you can see, from the start of the circle, there's discovery going on. Targets are discovered, drugs are invented and developed, and then they have to be preclinically tested. If it's good, it can go into clinical development and clinical trials. If it's really good and gets market authorization or approval, then there can be points for advocates to be involved in that post-marketing. And then even with pricing and reimbursement, that health technology assessments sometimes involve advocates to give feedback and so on. The print is really tiny in the blue areas, but it just goes into further detail on how all those time points, there's different activities you can be involved in. Next slide. And so this is also kind of an interesting issue that advocates can actually influence research community in a very broad way. And these two papers were advocate papers led by, spearheaded by Nick Bird, actually, and kind of looking at some fairly large topics. So when innovation and commercialization collide, really references what happens when a drug gets approved and is now on the market. There actually aren't very many incentives to further innovate because now it's a moneymaker. And this particular paper is primarily about the GD2 antibodies. And then the advocate view on transplant and, the debates that continue on, is transplant necessary? How many transplants in the era of immunotherapy? And so on. So that paper also really called for the research community to move quicker to less toxicity. And that came across loud and clear in the earlier session. So now I'm going to turn it over to Emily to talk more specifically about some research advocacy activities.
Thanks, Donna, and it's a real pleasure to be speaking in this session today. And just to give a little bit on my background, so my background is a scientific one, and I kind of moved into the research engagement field 3 to 4 years ago now, and since been working at Solving Kids Cancer UK, where I've learned a lot more about the world of research advocacy and what it entails. So just to start off by looking at some of the progress that's been made in this space. So more and more research networks are incorporating patient and parent perspectives into their organisations. So on this slide are 3 examples of where that's been really successful, but it definitely isn't conclusive. And there are other organisations internationally, such as NANT and others, that also have space dedicated for advocate involvement as well. So the Innovative Therapies for Children with Cancer is a European research consortium that's developing novel treatments for childhood cancer. In 2021, it established its advocate committee, and that now feeds into their executive committee that decide on the strategic priorities for the organisation. So a similar practice can be seen within SciAPN, which is the European Network for Neuroblastoma Research that we've already heard a lot about today. So its advocate committee was also established in 2021, and it has now been instated into the bylaws of the organisation to ensure that patient and parent voice is weaved into their work moving forward. So in both of these organisations, those committees are made up mainly of expert advocates that have had previous experience in similar roles, but both are actually looking to sort of extend out and develop broader advocate networks so that more parents on a wider level can get engaged at a more accessible level as well. So the final example on this slide is Accelerate, which is a multi-stakeholder platform working to accelerate drug development for children's cancers. This is distinguished slightly from the other two, as Accelerate was built to be truly multi-stakeholder from the outset. And as such, advocates have been involved from the very beginning of the organisation and sit right on the steering committee alongside representatives from academia, regulation and industry. So whilst it is good to see that there is a lot of progress going on, it's also important to note that this is an evolving field and there are certainly still hurdles to overcome when it comes to research advocacy. So it's not always the case that research involves advocates in the best way. Sometimes there isn't space for advocates to participate in research, or when there is, it can be tokenistic and doesn't always allow for meaningful contribution. Sometimes opportunities for advocates can have quite high expectations, so expecting individuals to speak on behalf of an entire community, expecting they have a certain level of knowledge on something, or sometimes speaking to knowledge that's beyond their own experience. And that's particularly challenging when it comes to children's cancers, because it covers such a broad field of diseases and even sub-disease types within that. So it's really important that opportunities to involve patients and parents are accessible, and that they take into account compensation for the time and sometimes travel that it entails for parents to get involved in advocacy activities. And this isn't always the case, particularly for those who aren't professionally involved in the field. It's also really important that individuals who want to involve themselves in advocacy have education opportunities. It's often the case that not quite enough is done to make sure that parents and patients feel that they are well equipped enough to speak on a topic that they're being asked to. Another really vital point to point out is that a lot of advocate positions are currently held predominantly by representatives from wealthier countries, and a lot more must be done to make sure that there's space for voices from other countries with less resource to give a more representative global view on research priorities and direction as well. And finally, for progress to continue in the way that it has, it's really important that we build up the next generation of advocates as well that can speak to today's landscape and kind of build on the work that's already been done. So just to expand a little bit more on that, research advocacy has very much developed in quite an organic way over the last few decades, as has the research that's being done. And what that means really is that at present, in a lot of spaces, there are small groups of what are considered expert advocates that work either professionally in childhood cancer organisations or have dedicated significant amounts of their time to develop that level of expertise. But really, for research advocacy to be truly sustainable for the decades to come that it needs to be, it's really important that we build a broader population of advocates with differing levels of involvement and expertise, and sort of move away from the all or nothing practice that we see a lot of at the moment. So part of doing this is to make parents aware of the opportunities that are available to them now, and also aware of their value in research. And hopefully this session today is doing some of the work to do that. But there is definitely also a responsibility for the research community as well to establish more accessible opportunities for advocates in the future. And for any parents in the audience who are keen to become involved in advocacy activities but don't really know where to start, these are a few ideas. So local and national charities that are engaged in research are usually well connected to the research community and might be aware of entry-level opportunities to get involved, or they might in fact have their own patient involvement forums or networks that you can participate in as well. So I mentioned that education is a really important part of parents being able to feel included in these things. And there are a number of online resources that can help to sort of give a better overall knowledge of the research and advocacy landscapes. So some of those are very beginner friendly, such as the UPATI Toolkit and the St Baldrick's Care Programme, whilst others have a sort of more in-depth focus on more complex topics. So the Aladdin programme is a good example of that and is really quite useful for parents who want to get a deeper understanding of a lot of the nuances on the landscape. So another point that Donna sort of alluded to as well is that for those interested in sort of immersing themselves more directly in the research community, there are often scholarships or grants available for patients or parents to attend scientific meetings, such as the ASCO meeting that Donna mentioned, as well as AACR or PSYOP. So finally, just to sum up, hopefully this talk has given you a bit of an overview of research advocacy, what it is exactly, what it can look like in practice, and also what the future might hold for research advocacy as well. And the important things to kind of take away from this is that it's a very broad field and it covers a lot of concepts and activities, and it is something that's constantly developing and improving along with the research that's behind it. And the other thing to note is that whilst we've established there are challenges in the space and there's a lot of work to be done to get parents involved, there are also a lot of opportunities for those of you who are specifically interested in research to get involved as well. So with that, I will say thank you from myself and Donna, and we both look forward to the discussion at the end of the session. Thanks.
Thank you very much, Donna and Emily. So next I'm going to invite Dr. Wendy London to come and present the Borneo project. Dr. London, over to you. I think we can't hear you.
Can you hear me now?
Loud and clear.
Fantastic. Well, thank you very much for your kind invitation to present at this symposium today. I'm very pleased to be able to tell you about the Borneo project, and I think this is an example of advocates advocating for research. We all know about the island of Borneo in Southeast Asia. It also happens to be a really nice acronym for our project, Biomarkers in High-Risk Neuroblastoma. So Lucas Moreno and I are leading this project, and prognostic biomarkers can be used to define subgroups of patients, low, intermediate, and high risk. And prognostic means that a particular factor is associated with survival. So children who have been defined according to biomarkers as having a high risk for a progression event or death are categorized as high-risk neuroblastoma patients shown in this blue curve. And until recently, all children with high-risk neuroblastoma received the same multimodal treatment. But it would be better if we could identify a subgroup of these high-risk patients who we could label as ultra-high risk and perhaps offer them a different therapy rather than the standard frontline therapy. So, for example, in the Children's Oncology Group, we use these factors to determine risk group at the time of diagnosis. So this is just an example of a list of biomarkers. So the idea that Lucas and I had is, well, what if we could get all the data from all known prognostic biomarkers, published biomarkers? That would be a pretty long list. But if we could get all of them together, we could do a comprehensive statistical analysis to compare the strength of these biomarkers and then identify the best combination of biomarkers to define which children are at the highest risk of dying. And so patients, parents, and advocates have been involved in this project, and they were involved from the very start, pushing us to transform this idea into a formal project. We're very grateful for the encouragement we received to submit an application to Solving Kids Cancer for funding. The advocates were understanding of the fact that there were risks with embarking on this project, the risk of not getting enough biomarker data or not being able to identify the ultra high risk population. But we appreciate that they were still keen to proceed because we all believe that there could be significant patient benefit. Another way advocates have helped us is with getting access to the data, and I'll tell you more about that shortly. So currently, we are not making use of prognostic biomarkers at the time of diagnosis to identify these children who are at ultra high risk. But if we had a group of biomarkers to identify them, we could give them early access to innovative therapies and potential changes in a treatment strategy. So to do this, our challenge is to get all of these novel biomarkers, put them together in one database, and perform an analysis. So the Borneo project has 3 phases. First, we want to perform a systematic literature review of all manuscripts about prognostic biomarkers in neuroblastoma. Then we want to obtain the data about those biomarkers and put it into the INRG Data Commons. The International Neuroblastoma Risk Groups Data Commons has been in existence since 2004 and is really the primary worldwide repository of clinical and outcome biomarker data for research purposes. And in phase 3, I'll, with others on the team, perform a statistical analysis to find a good combination of biomarkers to hopefully identify an ultra-high risk group of children. And then we'll take those biomarkers and validate their the definition in a cohort of children who have been homogeneously treated on a clinical trial. So at the Advances in Neuroblastoma research meeting held in Amsterdam in May, Andrea Villaplana gave a presentation about the results of the systematic literature review. And I'm going to show you some of the slides from that very same presentation at the ANR meeting. So the objectives of the systematic review were to find papers about the most strongly prognostic biomarkers. So published data, peer-reviewed publications on these biomarkers that were measured at the time of diagnosis. And we also want to collect from these manuscripts, published manuscripts, the definitions that have been previously used for ultra-high risk. So you can see in the pyramid on the right that systematic reviews like this are considered the highest quality of evidence and we want to use this systematic review to help us plan the next steps of our research to identify an ultra-high risk cohort. So systematics reviews build on randomized controlled trials and cohort studies and critically appraised individual articles, building finally to the systematic review where we take many manuscripts and combine the information to make our decisions. So we included publications between 1997 and 2020 about biomarkers from the time of diagnosis in neuroblastoma patients. The results had to have known data about survival, and we had to have a separate analysis within high risk and a sample size of 50 or more children. So we started out with a query in PubMed that gave us 5,829 manuscripts. So then when we further applied the eligibility criteria based on the abstract, 5,100 manuscripts were excluded. Some weren't even neuroblastoma. Some were only based on laboratory findings and not on patients, and some had non-significant results. And that led us with 726 manuscripts. So Andrea performed a detailed review of these manuscripts and further excluded 667 that did not meet the eligibility criteria, and then we ended up with 57 manuscripts in our review. These manuscripts, some of them included more than one biomarker, so we had a total of 68 biomarkers. 14 clinical biomarkers, things like imaging and procedures, 18 genomic biomarkers like DNA analyses, methylation, or telomerase, 4 circulating biomarkers like circulating tumor DNA in the peripheral blood, and then 32 protein or mRNA expression biomarkers. We used a tool called QUIPS, Quality and Prognostic Studies, to determine the strength and validity of the results in these manuscripts. So we looked at who participated, what the attrition was, what the prognostic how the prognostic factor was being measured, what outcome was being measured, was there confounding, and how the statistical analysis was reported. And so each manuscript got a grade, and a higher score meant stronger results of the prognostic biomarker study. So the results were good. We had high QIP scores with a median of 15, and some of the important things contributing to that high score is that 77% of the manuscripts were from multi-center studies. So that increases the generalizability of the results. And in 89% of the manuscript, the methodology for measuring the biomarker was well described. So this is a list of the 15 papers with the highest QIP scores. We're following through on all 68 biomarkers, but I just wanted to show you this short list. And more good news is that several of these biomarkers have data already present in the the INRG data commons, like ferritin and LDH. We also recently have added the Siopin and Curie MIBG score data. And of course, we have McCann already in the database, but we are actively pursuing adding additional biomarkers to the INRG, including those on this list. So in phase 2, this is where we really go after the data. So Lucas and I have contacted all the investigators and asked them to complete a worksheet with all the detailed information about where their data are located, what special permissions might be needed in order to get access, and who would be the right contact person to get the data. And then the next step would be for the investigators to provide the data set in an Excel spreadsheet or other means. The cooperative group statistician is really the key here, working with the investigator to de-identify the data, anonymize the data, or making sure that there are no protected health information like names will not be transferred. So they serve as an honest broker to replace all of the other identifiers, replace it with an INRG number. We have a unique identifier for each patient, and then the data will be deposited into the INRG data commons. So we had a very good meeting at ANR in Amsterdam, attended by most of the investigators whose biomarkers have been requested. There was a lot of good questions and enthusiasm to participate in contributing data. Having said that, it's still challenging from a logistical standpoint and just from the standpoint of making time to work on this project. These investigators are all very busy. And to that end, the advocates have really helped us. Solving Kids Cancer sent a letter to all 57 of the investigators. They were strongly supportive of the Borneo project, and they encouraged the investigators to participate by submitting their data to the project. So in addition, Lucas and I have emailed all the investigators with the worksheet and requested the data, and we've gotten very good response. Everyone wants to participate, and now we're just working on solving logistical and informatics challenges of the data transfer. So thank you very much for the impact that advocates have had in furthering the efforts of this project. So in phase 3 of the project, we'll perform the statistical analysis to find the best combination of prognostic biomarkers to define the ultra-high risk group of children. So again, these are children who are likely to die if they're treated with our standard high-risk therapy and could potentially benefit from a different or novel therapy like a targeted therapy. So a hypothetical example to define a ultra-high-risk group would be if we had patients who were over 18 months at diagnosis, had metastatic disease, their tumor had MYC amplification at high LDH and 11q LDH. So for example, we want to find out what those biomarkers are that would allow us to make this definition. So how do we pick the right combination? The best example of the methodology that I plan to use has been shown in our paper in Journal of Clinical Oncology for the INRG risk stratification. So this will use recursive partitioning, where we take the overall cohort and repeatedly divide it into 2 groups, based on a particular biomarker. So for example, we could compare the outcome of younger children versus older children. And using a Cox proportional hazard regression model, we'll calculate the hazard ratio, and this quantifies the difference between the survival curve for younger children versus older children. So age is one example of biomarker that we can use to create these two groups to be compared, and we're going to do that over and over again for every biomarker, and then we're going to pick a biomarker that has the largest hazard ratio, and then we do it all over again within each of the subgroups that we've created, and that's what I mean by recursive partitioning. So one of our biggest challenges is going to be the fact that we have missing data. So shown here, if we're, for example, looking at two biomarkers, A and B, so we have in blue known data for biomarker A and some known data for biomarker B, but within a particular group of patients, there might be only a small overlap where we have known data for both biomarkers. And then also we have a lot of data that's unknown for biomarker A or biomarker B. So I'd be happy to discuss with anyone outside of this meeting the details of the propensity scores analysis, but this is an approach that's going to allow me to estimate the value of a biomarker if it's missing, and hopefully we'll have a much larger cohort of children with known data for all of the biomarkers so that we can do these head-to-head comparisons. So the next step is for us to submit the systematic review manuscript for publication. We've made good progress in adding data about MIBG scores, the Curie and the Sciapan scores, and the next data that'll be soon added to the INRG are on ALK mutations, response to induction, and some novel genetic aberrations. We're working to improve our access to data from public databases like the Target database in North America and R2 in Europe. I can't thank Solving Kids Cancer enough for the support they have of this project. I'm very passionate about this project. I want to thank in particular those that I know personally, Donna Ludwinski, Nick Bird, Leah Ambler, and Leona Knox. I'm sure there are many more names that are missing from this list, and I'm sorry about that, but I'm sure you're working. We're all working very hard. I especially want to thank the patients and families who have participated on clinical trials, who have contributed their data so that we can do this critical research to help children in the future. I want to thank the Borneo team, Andrea Villaplana, who's worked on a systematic review. Paige Cow is a statistician in my group who will be working on the analysis with me. And of course, my good friend and colleague, Lucas Moreno. I want to thank the statisticians at the cooperative groups who are going to be working with the investigators to submit the biomarker data to the INRG. I look forward to discussing the Borneo project with you some more. I think it's a great example of a way in which advocates have helped further the research agenda in neuroblastoma. Thank you.
Thank you very much indeed, Dr. London. And we all very much hope that the Borneo project will lead to some interesting results in terms of trying to better stratify children prospectively at diagnosis. If anybody has any questions on the Borneo project or for Dr. London, then please pop them in the chat. We will have time for them at the end of this session. But next, I'd like to introduce Professor Maya Beck-Popovich, who is going to lead the next segment on neuroblastoma in 2035, perspectives on the direction of research. Maya, it's over to you.
Thank you very much, Nick. Hello, good afternoon. Thank you very much for giving me the possibility to participate in this very interesting discussion. And also on the role of patient advocates in research. Now, we have heard since the afternoon started many interesting topics. Almost all or many aspects in neuroblastoma high-risk patients have been covered by the different participants. So I think that I will try to make like a summary on where we stand and have a reflection then with you together, or where we shall go next. So many of the topics also concerning futures have already been discussed, but I think it's good to have an overview of progress we have made, maybe of knowledge we have acquired, and what still needs to be done. So I have nothing to disclose. Already at the beginning of the afternoon, Dr. Kohn has nicely presented one big step that was needed and that had to be done in order to have our community taking care of patients with neuroblastoma talk the same language, that we can know when we discuss a patient and problems related to that specific patient, that we know what we are talking about, what stage it is, what kind of disease, different aspects. So this, among the progress we have made over the last decade, certainly one important step, that worldwide we can say today we have a common language. When we talk about staging in neuroblastoma patients, we know what we are talking about. We have developed a common classification system that allows us to have the same definition for low, intermediate, high-risk patients. And as we heard right now, we're going to have to develop tools in order to define ultra high-risk patients. Very important also, we have over these years not only reviewed and agreed on how to classify patients, but we have also improved quality and harmonized and standardized how we should do some analysis, how we do pathology evaluation, how we do biological analysis, molecular analysis, what are the needs for laboratories, what are the standards that centers need to have in order to be able to have correct biological results. So the quality assessment of pathology and genetic markers is another common step that we have done in the last years. And then also, when treating patients, we need also to know how we do evaluate response to treatment. So to have a common way of evaluating and knowing how disease is responding to treatment is absolutely necessary in order to be able not only to develop new treatments, but also to know how do we evaluate response to treatment, standard treatment or new treatments in evaluation. We have worked on surgery. Surgery is important not only in localized disease. We have seen that an important role also, surgery takes an important place also in patients with high-risk disease. And as it has already been pointed out earlier this afternoon, is that with the help of surgeons and also their enthusiasm in order to know when to operate and not to take unnecessary risks that might compromise further treatment, be it chemotherapy, be it immunotherapy or any kind of radiotherapy, and not to operate when risks are too important. So with the help of surgeons and radiologists, this pre-surgical evaluation has been developed and implemented now. We can say worldwide, we use the same criteria in order to evaluate whether a patient can be safely operated or not, or where it is much more reasonable to have a biopsy done and to start with chemotherapy or other treatments. Then very importantly, and this has also been in many ways discussed this afternoon, biology. We have made progress and acquired knowledge in a very extensive and important way. in addition to NMIC amplification, which is certainly one of the most oldest and well-known biological prognostic factors, we know now that there is a world of abnormalities that we find in genes, that we have many aberrations we have to consider and to consider and as risk factors, and also to implement in our reflections on treatments. We know also that and different groups have worked on in Europe and in US also on telomerase, and as you see on the slide on the right part, on telomerase maintenance mechanisms, which is another we can say world of biological abnormalities that play a significant role in prognosis, and we have to consider when we think of treatment, when we develop new treatment modalities. This is an overview on trials that have been done over the last 20-25 years. Now, these are SciAPen trials. We have heard about CRG. In fact, many of ideas are overlapping, and certainly in the collaboration, international collaboration, which is another important aspect that has been also much reinforced over the last 20 years, the aim is that more and more we do research together, or we ask complementary questions and not same questions just repeated here and there. So the intention here is to show that over the 20-25 years, the last decades, we have had various trials, be it on localized neuroblastoma, localized but unresectable disease, neuroblastoma in infants, very long-lasting high-risk neuroblastoma protocol, and as already some of the precedent speakers have pointed out, various questions have been through randomization also answered in this high-risk trial. The current ongoing second high-risk trial within SciAPen, we had presentations in information on relapse or treatments in relapse patients. We have trials that tried for patients not responding well to induction treatment, intensification of treatment, and then also in other patient subgroups that are not high-risk patients, but important patients like children that have opsoclonus myoclonus syndrome or children that have spinal cord invasion. Also ongoing trials that try to answer different treatment aspects in specific settings. And then another important development, which is very much in line with the overall effort to have put data together in order to have a lot of information that we can use in order to evaluate and to find out not only prognostic markers, but also to know how neuroblastoma patients are doing, also those who are not on trials. We have now the bioportal that European-wide will collect data on neuroblastoma patients that are or are not on trials, also with the aim not to lose any kind of information, that we really have a maximum of information on all patients that develop neuroblastoma. So this is a busy slide, but in fact, many things have been presented already this afternoon, so I will not go into details. This shows just for the high-risk patients what we have acquired as knowledge through randomization. So we have within this over 20 years lasting trial information on what is in the SciAPen setting the best consolidation treatment. We have compared various types of induction treatment and have for the moment rapid Kojic as the standard treatment. But as you might know, there is again a randomization in the current high-risk protocol of SciAPen. We have implemented anti-GD2 as soon as the results from our US colleagues have been available to all patients, but then asked the IL-2 question in a randomized way and have then concluded that IL-2 did not really improve results, but had some toxicity, and this has then been even important toxicity, and this has then been dropped. We have then also answered questions or had this. This has also been shown by Daniel Morgenstern at the very beginning, that infants with anemic amplification and high-risk disease really had in the infant protocol unsatisfactory results, bad results, and do much better on this high-risk protocol. So many of the questions that have been put like 20 years ago have been answered in the meantime, and are implemented, most of them now, what is the high-risk 2 protocol. So if we look, and this has also in some parts been presented, this is what the treatment that is currently the trial that is currently open within SciPen for high-risk patients. And you can see it is an intense treatment. We have the induction block with a randomized question on 2 types of induction treatment, but we have a treatment intensification for patients that are insufficient remission at end of induction. We ask in the SciPen setting the question for a subgroup of patients between 1 versus tandem high-dose chemotherapy, high-dose transplant. We also ask a randomization question for patients who are not in remission at the primary tumor between 2 doses of radiotherapy and have then a standard treatment for maintenance. So if what you have heard the whole afternoon by the different presenters, and when we see what the information and the knowledge that has been acquired over the last decades, we have for high-risk patients clearly increasing treatment intensification. Be it because patients are not in sufficient remission at the end of induction, or because they progress during treatment, or because they will later relapse. And there has been also a whole topic on how relapse patients, whether we have a standard treatment, what are the ways of giving treatment to relapse patients with it, which is a topic in itself. So how can we look at when we look back? We have improved results, but if we take all the patients together, we still might cure 50 to 60% of high-risk patients, but this is certainly not enough. We also have, and must not forget, low-risk and intermediate-risk patients that are not all cured. We know that there are also subgroups that need different, either intensified treatment or different treatment modalities, because they will not be cured. We have increased knowledge, we have more and better tools, we have developing collaboration worldwide, but we have also increasing needs. We need new treatments, we need new treatment approaches, and we need time. So this is an important component. We need time, but we know that we cannot lose time and that we have to accelerate. We need to be sure that treatment is accessible to all patients, be it in a first-line setting, be it in the relapse setting, be it also in the second or third relapse. We have not yet a standard approach to relapse, and we know that this treatment intensification has also costs on the other side, on toxicity for patients that are cured, on possible sequelae, and that we need also a very well organized and careful follow-up of patients that are cured. And we have this international collaboration that certainly is going the right way, closer and closer, doing more and more together. And this opens new doors, because together we are stronger and might gain in time. So at the ANR, and there have been presentations of highlights from ANR, and the aim is here not to repeat highlights, but we had the opportunity, a very interesting session with patient advocates. And the idea of the discussion was, independently of who we are, from which collaborative group we are, to sit together and to see what are ideas, what is important, not only to doctors, scientists, but also to parents, to patient advocates who have their own experience, who have their ideas, who have needs, to discuss and share how future should look like. Where are we going? What are the important aims also from a patient advocacy perspective? And I think what Wendy London also has shown, where the input from patients can come and advocates, is in many, many ways. And one important input is also to know, how do you see, where do you see neuroblastoma research has to go to? What are the important topics that we do not want and not have to forget? Because I think that as doctors and scientists, we certainly do not claim to have all the knowledge and all the thinking of all the important factors. So these were, and I think now Emily and will take over, Emily and Donna, but these were the questions we have asked at ANR, and it is a good opportunity to have at this moment also your input. So I will stop here. Thank you very much.
Thanks, Maya. Yeah, so as Maya said, this was a really successful session at ANR, and we really just wanted to be able to recreate some of the similar discussion that took place, but within the parent community. So hopefully on the screen now, you'll be able to see a QR code, and this is the program that we're going to be using to get your audience participation today. So we're going to be running through each of these questions, and to get access to participate, if you either just scan the QR code or go to slido.com and enter in the participation code, and then you'll be able to join. So these details will be up on each of the slides. But what we'll do is we'll just run through each of the questions, and then I'll hand it back to Maya to kind of facilitate the discussion of what comes up. So first of all, just to get everyone familiar with what they're doing, just a nice easy one to start with, about where you're all joining from today. Okay, great. Looks like we've got people in and logging on, which is brilliant. So I'll move on to the first question that we'll be discussing, which is, what's the most exciting advance on the horizon for high-risk neuroblastoma? And we can see some answers coming up already, but please do add in. And even if you've got an answer that you can already see, do add that in, and that will help us see which ideas are more popular. Okay, it looks like we're getting quite a good spread of answers. So these questions will pop up for you again as we go through the discussion. So I'll just move on to the next one for now, which is, what is the priority to improve cure? see lots of people still typing on this one, so I'll just leave it for a couple more moments. Okay, and then we'll move on to the final question, which is, what is the biggest challenge to achieving the goals that we have? Okay, brilliant. So as I said, these questions will pop back up for you as we move through the discussion. So if you want to add anything as we go, you're more than welcome. For anyone that's not able to participate in the Slido as well, do feel free to pop things in the Q&A box as well. So Maya, I'll hand back over to you and we'll look at the first question and the responses.
Yeah, thank you very much. So I think it's very interesting to see on this slide the most exciting advances. It fits very well into the treatment intensification we have seen through chemotherapy. What I can hear and see is that all these treatments have two parts we don't have in the classical chemotherapy. First, it is more directed to disease specificity. Secondly, it is treatment that has that is less toxic, that is not without having adverse events, but that with the knowledge we have now might be not as permanent and not as deleterious as chemotherapy sometimes can be. And the other thing is also in terms of liquid biopsy is also less aggressiveness, because liquid biopsy not only might help to avoid some surgical interventions that we need for diagnosis, but it might also help in the follow-up to have and develop tools where we can, through a simple blood sample, follow disease and maybe do less bone marrow analysis and bone marrow investigation for follow-up, radiological, etc. So I think it is like a more specific targeted treatment on one hand, and on the other hand, tools that are less aggressive for diagnosis and follow-up of patients that already have a very heavy burden of treatment. I was, if I can say, to what I picked out, I'm not sure if it's here or in maybe in another slide, but it is in a way linked to that. We need, of course, trials in order to evaluate these new treatments, to evaluate not only their toxicity, to evaluate efficacy, and we need for this protocols. And somewhere there has been a remark, more protocol flexibility. So this is very interesting, because what is exactly meant with protocol flexibility? Protocols usually they have to ask questions. There is a way to answer these questions. For this, some assessments have to be done, and they also give usually rules how to behave according to response accordingly to toxicity. So it is quite strict, but I just thought it was an interesting, and I would be happy to hear what was meant exactly by that. So I think that certainly what is here on this slide, this is a priority that is very much in line what the current priority is. And we have heard about immunotherapy CAR-T in relapse and liquid biopsy. There is a big project ongoing in preparation, also European-wide, also in US. And I think this is really the way it is going.
Thanks, Maya. And just moving on to the next slide, we can see actually the responses that we had from the poll at ANR, and I can see there's a lot of overlap there between what the research community and what the parent community are saying, which I think as advocates is really encouraging to see, actually, that we're all excited about the same things at the moment.
Yeah.
So moving on to the next question, which was, what's the priority to improve cure? Again, lots of similar things coming up in this one.
Yeah. Yes, I think when we are lucky to have really targets that we can use also. There has been earlier this afternoon the question about 11q. So not all the molecular abnormalities we can find are automatically a target. So when we are lucky to have targets, of course, and to have also drugs against these targets, it is certainly the combination and implementation into treatment an important step forward. And we are lucky with ALK, with lorlatinib, although it is a minority of patients who have this. But if we are doing also these worldwide projects on molecular analysis in order to understand better what important biomarkers are, because there are so many, and so many researchers are working on prognostic markers, it is really important to have an overview in order to know what really makes a difference in prognosis and what can also be used then for treatment development. So I think there is also concordance with what we think is the future, and this is targeted therapy that has to be added in one way or the other to some background treatment, but certainly it would help not to increase intensity by increasing also toxicity, but increasing by modifying, improving the quality of treatment we are delivering.
Thanks, Maya.
Sorry, excuse me. I was just ctDNA monitoring. Of course, this is very important because this is the question for the moment. We have really gross tools to realize when there is a relapse occurring. What we wish is to discover the relapse when we cannot see it yet. And this is certainly ctDNA monitoring, and that's why also the Mona Lisa project is an important project that will help to ask these questions. But we need to have markers that can detect when relapse is starting before we have already the gross tumor or even metastatic spread in the relapse setting.
Yeah, absolutely. I noticed one of the other big ones that's coming up here is information. And Donna, I know you kind of mentioned that in your personal experience, that was a huge thing going back decades ago. So I don't know if you wanted to comment on that at all.
Well, yeah, that's a really good point. When when he was first diagnosed, there was no PubMed, there was no clinicaltrials.gov, there was no anything there. You really couldn't find out anything. You had to depend on, I mean, if you wanted to go to the library and look at microfiche, there wasn't a whole lot there, but that was it. So now, of course, the flip side of so much information for parents is, there's no going back. In other words, you can be overwhelmed. You can be terrified by what you read. It can be, it can be quite onerous. So, there's definitely a downside to so much information, but again, there's no going back. So I do think, though, what was brought up earlier about the efforts to, prioritize trials that make sense to try to get to what Dr. Moreno was talking about, I thought is really brilliant. I think there's a huge need for that, because then there's a trusted source, and I think that's exceedingly helpful concept.
Can I also add a comment to that? I think there is also an important role of advocates in information. And I think that meetings, in my experience, when meetings are with participation of advocates, it really helps then also to diffuse information and also to help to pick out the correct and the good information. And I think this is also one of the roles, because as you have said, there is so much just accessibility, but not all information is useful. Some information can even be dangerous. So having a clear list of trials that are ongoing, of also new research, and having then also help from advocates to distribute information, to organize meetings where this can be discussed and updated is really very important, I think, and helpful.
You know, I'm glad you brought that up, Maya, because I was thinking about this interesting phenomena with patients being so easily connected with, global forums. And I remember in the early days of antibody therapy, parents would post, "Hey, has anybody ever seen one pupil of the eye being bigger than the other?" And these conversations were happening before clinicians really had a chance to aggregate that kind of data. I mean, an individual clinician would think, well, that's strange and we don't know why, but didn't know that was happening necessarily widely.
Yeah.
And there's plenty of examples like that I think if we could harness some of this real world data, it might
Yes.
be very, very useful. I was thinking, for example, the phenomena of MIBG positive disease that's PET negative. Seen numerous anecdotal evidence of how do you interpret that? What do you do? But it's a thing, and it's discussed quite commonly. So that would fit into the whole, response criteria. Maybe that's a question for the future of what to do in that setting.
Exactly, maybe summarizing also or having a session specifically on observations of parents' observations and not knowing whether this is important or not. I think this is also something that can help increase information for all, and certainly also help to say, well, this is something that we have to work on because it happens so often that it must mean something, and we have to try to give the correct interpretation to that. So yes, clearly.
Yeah, both really good points, and it does just show there's still so much more to be done in this space in terms of advocates and researchers working together. So again, we can see the results from ANR on this question, and again, lots and lots of overlap. I'm just going to quickly move on to the next slides that we can address the last question before we move on to the main discussion, which is what's the biggest challenge to achieving our goals? And I think this one is maybe more easy to discuss and more obvious to all of us. But Maya, I'll hand over to you.
Yeah, of course, this is very important. Now, how shall I say? There are different types of funding because there is funding for a trial, and depending on what the final aim of the trial is, whether it is a really research question, whether it ends in approval of a new medication, of course, the needs are different, and the whole project, in a way, is then different and depends very much whether it is an academic project or a pharma-guided project, and the funds are different. This is one aspect. Another aspect, and this is maybe one of the barriers we have very often, is then per country, the opening of trials. That additional funding is needed, and sometimes this, so many times, can just delay opening of important trials and participation because this funding is per country is a problem. So I think that Lucas Moreno has mentioned earlier that we have, and it will be an obligation for the whole European community and all the European countries to be on the CTIS platform, which is then a European platform that should help have a more standardized way of opening trials and the same rules for everybody. And it should be helpful to accelerate opening of trials. It will not resolve the funding. And then, of course, I think the role of patient advocates, of course, is also very important in this setting, but it cannot be only advocates. So systems like Accelerate, putting together patient advocates, pharma, academics, So all the necessary participants in research, maybe that this is the best way to go, because to have an open discussion on where we stand, what is the priority, and if we decide that a trial or a scientific question is a priority, or a new treatment that has to be evaluated is a priority, then we should really also agree and fund this trial. So there I see also a very important help from advocates, not in the means of fundraising, but helping to make it understand that if something is a priority, then also authorities and politicians should agree and help.
Yeah,
So this
absolutely.
is
Donna, I don't know if you've got anything to add there.
That's a perfect summary. I was thinking about, I had an opportunity to attend a meeting in Canada fairly recently that was discussing these systemic issues, because Canada is a, relatively small country by population. So opening, every trial on earth is impossible, and how to prioritize and what are the obstacles and limitations. And fascinatingly, one of the issues that came up was just the time it takes to accomplish contracts. That, there should be templates for contracts and that sort of thing. And it's such a practical solution. And it reminds me of years and years and years ago, I was talking to a neuroblastoma expert in the UK who said that he believed that conflicting layers of regulatory issues end up, creating cycles that waste time. And obviously, time is of the essence. And so, I never really thought about those, kind of behind the scenes issues, because as an advocate, you're not necessarily involved in those kind of things. And then a prominent German doctor once, he coined the term regulatory related mortality. And I thought, oh, that's terrible to know that, the systematically there are maybe unnecessary complications that could be addressed.
Yeah, I think you're completely right. This is really a very important aspect to be added. And we might have some lawyers here that are knowledgeable in the field that are listening, but there are very, very precise examples, and it can be so different from countries. And I know that our German colleagues that need so much time for the contract and all the aspects, and it's a ping pong. And this is also with others. This is just an example because it's a very recent example, and we have a lot discussed on that. But it happens also that each country asks for something specific. And if you, like Switzerland, are not part of European community, it then, in addition, complicates the situation. And it should be just one language for everybody who is in Europe. It should be the same. So, but maybe I don't have a lawyer's mind in order to understand why the difference has to be so big and so important between the countries. But this is also a reason of delay, clearly.
Yeah.
Thank you, Anna.
Thanks both. Both really good points. And again, we've got no surprises really that the responses from ANR, we can see funding right there in the middle again, and time as well. So just conscious of our time now, I'm going to stop sharing my screen. I think we're going to welcome the rest of the members back to the stage to start the general discussion. Oh, we can't hear you, Nick.
Thank you, Emily. Saying thank you very much to the three of you for taking us through that, and very interesting to see such similarities between what we had in the polls at ANR and what was received today. So we do have some questions. So question, I guess, let's give this one to Donna to kick off with. Is the difference in awareness between breast cancer, etc., I guess, and pediatric cancer due to advocacy, funding, etc.?
Well, certainly magnitude would be, a huge difference. You just look at the number of breast cancer diagnosis, the number of researchers, the number of patients that are involved in advocacy. They actually have an army, and I think they've really kind of elevated the concept of research advocacy as a disease group. That it's going to be really difficult for us to really kind of achieve that magnitude, just given the numbers. The fact that they're dealing with roughly one disease, and we're dealing with so many in the pediatric cancer space, obviously is a big factor as well. And just the amount of money that they can bring, and the amount of influence they can bring because of the money and patience that they can literally bring to the research is it's just on a whole several orders of magnitude more than we can do. But we can certainly look at principles of what they've done and their successes in order to try to emulate that in our sphere.
Yeah, so the orders of magnitude, there are over 2 million new diagnoses of breast cancer a year. And, that compares to neuroblastoma in particular, where we've got only 700 new diagnoses in North America. And just like you said, I think the biggest difference here is just sheer numbers.
Mm-hmm.
So maybe I can ask a corollary to that question then. If it were sheer numbers, one would expect the pediatric cancer advocacy landscape to be dominated by acute lymphoblastic leukemia, but at least to my mind, it's not. So I wondered if anybody had any thoughts on why that might be.
I don't know.
Or maybe I'm wrong. Maybe it is, and I'm mistaken.
I've heard comments over the years from researchers who deal with many pediatric cancers saying that the neuroblastoma community is different and nobody knows why. In other words, just the coalescence and the connection. And I mean, I think the community is different, but I don't know why. And I also don't know why the community of researchers is so top-notch, so numerous, so interested in a disease that, at least in the high-risk space, that's only 3% of all pediatric cancers, right? So why does neuroblastoma attract the attention of the brightest researchers, and why is it that the neuroblastoma community is so active and recognized? I don't know the answer to that.
Nick, I didn't understand well your question. You were asking whether the leukemia community is more active or more important compared to neuroblastoma, whether neuroblastoma is the most active one.
Yeah, whether or not, given there are obviously more patients, more families affected by leukemia, that actually one might expect there to be more advocates in the leukemia space.
Yeah. Maybe to addition to what Donna has said is that there is it might be a question of numbers, because disease is much more frequent than neuroblastoma, and research is progressing more quickly because there are numbers, and we have the difficulty with small numbers in neuroblastoma, and even smaller for these very high risk patients that do not do well, and this then needs people that invest time, effort, interest in order to improve that. So maybe this could be an explanation. And then, of course, it is such a heterogeneous disease. It is such a so different aspects, and leukemia is molecularly also heterogeneous, but maybe on the whole a little bit less than a neuroblastoma. Some thoughts.
Wendy, I'm going to come to you with this question. On the topic of biomarkers and prognostic factors, has the view regarding endemic amplification changed at all during the last years especially for children that are already stage 4/high risk. Kids that make it to the end of therapy are still considered greater risk for relapse than non-amplified or not?
This is an excellent question. And in fact, we are doing 2 studies to answer these questions. We have done a lot of prognostic studies based on data prior to the era of the introduction of antibodies. And we need to repeat our studies now in the era of these more modern therapies. So we have one study that's going to look at the changing role of biomarkers over time and the improvements in outcome over time. And also, and that's with Boris Dikarolis. And then I'm working with Daniel Morgenstern to redo an analysis about what factors are the most highly prognostic starting from the time of relapse. And so to my knowledge, we have no evidence right now that there has been any big change in the role of MYC amplification as a prognostic factor. But if there is, we will find out with these 2 new ongoing studies, hopefully published next year.
So I'll ask you this, but you've kind of answered it already in what you just said, but it was an object of research in Borneo to also identify biomarkers that cause early or late relapses, as well as measure of aggressiveness based on the biomarker.
So at this time, we are using a methodology that's going to make use of a survival curve. So it looks at the entire length of the curve. At one point, Lucas and I tried to categorize early versus late relapses, and we found our results were misleading. And so instead of using a binary endpoint like early relapse or late relapse, we're sticking with the whole time to event endpoint, which has results that appear to be more intuitive. And so I think the whole concept of trying to categorize the timing of relapse, it may be useful in predicting overall survival post-relapse, but not as a biomarker necessarily to be used to stratify treatment.
So I'm just going to come to you again, Wendy, so you take yourself back off mute. Just one more. This is a question, obviously, that we as parents and advocates always ask, is when can we expect the first results from Borneo?
I think it's going to depend on how rapidly we can assemble the data. It's the practical issues. As a scientist, I've got my analytic plans ready to go for the statistical analysis. We've been thinking about it a lot. We've done a lot of programming. The thing that's not ready is the practical logistic issue of the data. I would hope that we would have, I don't know, we're going to work really hard to get the data as quickly as possible. I would hesitate, and I'm not giving up this project, no matter how long it takes. I know we have timelines that we're going to work very hard to stick to, but if we find that some of our key biomarkers are going to take longer to get enough data, then I'm happy to keep working on this until we can get all of the right data. So I hesitate to answer that. I would hope that we would have some answers by the end of next year. I'll go out on a limb and say that.
And we'll certainly help you to get there if at all possible. That's for sure. That's Emily and Donna. I'm going to come to you with this one. Is there a clear pathway for potential research advocates to gain the necessary knowledge and expertise? Can one be an advocate whilst also holding a full-time job? What training would you recommend?
I think the question could be for Mr. Nick Bird.
Yeah, I said I could certainly open it up, but I would expect this one to come back to you, Nick. The easy answer to one of those questions is yes, you can definitely be an advocate whilst also holding a full-time job, and Nick actually is an example of that. But also, what we hope to see is more and more opportunities coming up where it doesn't mean that someone has to dedicate all of their time or their career to holding, advocacy positions. In terms of a clear pathway, I think it's hard to say yes or no on that one, really. I think in terms of the advocates that we see in the space now, a lot of them have sort of evolved very organically, and a lot of the time have had to, fight to get a place in rooms and to sort of educate themselves. So I think there's definitely improvements, and obviously in the opening talk we did highlight some of the resources that are available to take steps to move into this space, but there is also a long way to go, I think. But yeah, I'll definitely defer to Donna and Nick to speak a little bit more about their experience in that as well.
Yeah, I guess, Emily, you summarized that so nicely. I think you start out not knowing what you don't know, and that makes it difficult to figure out what you do need to know. That's why these training programs are so, I think, so amazing, because even going through those, you'll stumble across something that's like, wow, I need to know more about that thing, be it regulatory science or, just drug discovery, or, how industry, how they do their business calculations on what they're going to, pursue. But I do think that it doesn't hurt to start out, with something that's a passion area. So one of the first things I did was apply to be an FDA patient representative, and the training was actually fabulous. So it did a dual purpose. I understood more about the drug development life cycle, but I also understood more about the function of the FDA. And so then that sprung other opportunities. I applied to be on the central review board. So being on an ethics board is a totally different role, right? You're looking at, kind of the risk benefit of trials, that's kind of the last stage before a trial gets open. And so learned an awful lot there. And now the NCI has a steering committee that considers early concepts for phase 2 and phase 3 studies for the COG. And so just recently I got on there. And so now that's a whole different role. So sometimes getting in the role helps you understand kind of the inner workings, and then identifies more things that you need to know. So it's really kind of finding the passion area of, what appeals. I mean, we talked earlier about the contracts. If there were lawyers in the room that could help develop templates for contracts, that'd be great. Be amazing. That's not going to be something I can do.
Right. I mean, I very much started out in the UK before I ever became involved internationally. And I think if there are opportunities for parents to get involved within their kind of national research bodies or local institutions, then it's just an opportunity to dip the toe in the water and kind of just see and start to get a feel for being involved. Let's take another couple of questions. There's a question on treatment here, Mayra. Why does immunotherapy not always work if the tumor has the GD2 receptor? So I guess this is the question around the fact that, everybody says that GD2 is abundantly expressed on neuroblastoma cells, and we give children anti-GD2 antibody therapy, and yet for some children it still does not work.
I think I cannot answer precisely. This is Juliette could do that in a much better way, but there are escape mechanisms clearly, and this is also something that will be addressed, whether a combination of different types of immunotherapy, not only anti-GD2, but like also anti-GD3 might be, and other receptors should be targeted at the same time, because there is in general no treatment whether there is not at some moment resistance or not response. So this means that the cells do escape, so they can express, but then another mechanism will just hide the receptor and not make it accessible. And this is also a field of research, and to see how in these patients then other receptors have to be targeted, and this will help to have the novel models of immunotherapy that combine different receptors in order to make them efficient also when one of them escapes response.
I think this is going to be a big focus on the next upcoming COG study, to actually try and figure out which children respond to chemoimmunotherapy and which children disease is more resistant to it. There's a comment here about ALL, which actually I kind of agree with, that the ALL treatment is more straightforward and probably much more protocol driven, even in the case of relapse, which probably lends itself to the kind of parent advocacy, given that neuroblastoma is very complex and there are multiple pathways, and parents in relapse refractory situations are involved in decision making along with their clinical care teams. Another one here on advocacy. How do we as parents bring our skills to the table to help move things forward? Maya, you gave an example of needing lawyers. How do we learn what the issues are and get involved?
Oh, I would contact the national regulators, because you have the ethics committee. This is usual, depending on countries, it can be very local, it can be national wide, but there is always a national organism which is really the deciding body on the legal level. And I would start there, because just to give you another example, Switzerland is one country, but we have all these cantons, and law is different, and the regulations from one canton to the other, it's not exactly the same, but there is a national organization, and I would go there and already try to find a basis for national wide. And these are then people that can be connected then with legal organs in other countries and European wide. So I think that in this way, that this is how I see how involvement could be possible.
I guess my comment to that also would be, in Symposium, obviously under your leadership, Maya, there's now a advocate committee within Symposium, which is headed up by my colleague Leona Knox, and we certainly would welcome anybody getting in touch who could offer any particular skills, etc. Not everybody who is involved as an advocate will have the same time capacity, but certainly if a broad range of skills, we can certainly help in many different ways. Donna, anything to add? Emily?
No, I think you covered it well. I do think it's. I think maybe the answer is just pick something, start somewhere. Like Maya said, start with a place where you can get involved, have conversations, and learn, and then find where you feel like there are unmet needs and a place to plug in, because there isn't a formal path. Emily will put that it's truly been an organic experience, and as time goes on, I'm hoping that it'll become more, that smooth path will be provided. But there are just numerous ways to get involved in, and who knows what's the most satisfying to an individual until they get in that space. So.
And I think unless you're super committed, just start somewhere where it's a safe space, and you can withdraw if you find it doesn't work for you. So just find those things. Wendy, I'm going to come back to you with the next one, the next $64 million question. Even if the Borneo project is successful and identifies very clear prognostic biomarkers, does Dr. London see this work being put into clinical practice quickly?
I hope so. So the way we would proceed, in COG, I can tell you over the last 25 years, we have changed the risk stratification, big changes twice, right? So when you make a change like this, where instead of assigning a patient to a group where they get a particular protocol-driven therapy, now you're saying, okay, you're now going to be classified as ultra high risk. So this is the same as testing in experimental therapy, because we are now changing the frontline treatment for a group of patients because we have reclassified them in terms of their risk group. So this has to be tested prospectively the same way we would test an experimental drug. And so we would make the change to the risk stratification. We'd have to test it in a trial. So you're going to be testing two things. You're testing the fact that a patient is reassigned to something different than what they would have gotten in their original risk group, the high risk group. But you're also, within that ultra high risk group, going to be testing a novel therapy. So it's going to be hard to distinguish between does the novel therapy work versus how have we affected the outcome of these children by reclassifying them. But what I've just described, I hope that once we have the information from Borneo, that we can proceed both in North America and in Europe to revising the risk classifications to provide this opportunity for these ultra high risk patients to have access to the novel therapy. I think they're the ideal group to both benefit from a potential novel therapy and both provide the most appropriate group of patients in which to test a novel therapy. I hope that answered the question.
It sure does. We have a question here. Maybe we'll put this out for others to answer as well, who were on the clinical session earlier today. Are there any? So obviously, in all of the major centres, induction therapy is multi-agent chemotherapy given in blocks. There's a question here about whether there's been any plans or discussions to investigate the combination of irinotecan, temozolomide, and dinutuximab in upfront induction therapy for newly diagnosed patients. Which I guess, to my knowledge, there hasn't been any firm plans, and maybe that's something to do with with how strong the chemotherapies are. But I don't know if you want to add.
Yeah, I've seen this question. I wondered why exactly this as induction treatment, because it has to be proven that it's really as efficient in relapse that it seems to be. And chemotherapy, there are plans to add it to immunotherapy to other induction backbones. This is what the final aim of the current high risk study is. So I wondered why this combination in induction.
So I think it's probably driven by the fact that temozolomide irinotecan is more well tolerated than the multi-agent chemotherapies that are given in induction, and maybe doesn't have the ototoxicity and the short stature and those things that are coming out of the LEARN study, Lisa Diller's work in the US. And given that it's the combination that's being used in the relapse refractory setting in COG, that whether or not there were any plans to evaluate it in frontline.
Can I just make a comment to that? Irinotecan is quite toxic. It is on the digestive level. It is not always well tolerated. So I can understand the question to have maybe a less toxic treatment. But compared, I must say there are also other induction treatments that might be with well tolerable combined to immunotherapy. So I think it's an open question.
I've just put in the chat a recent publication by my colleagues at St. Jude. It's not irinotecan and temozolomide, but it is a trial that's looking at putting dinutuximab in upfront high-risk therapy.
Thank you very much. So I think we've got through pretty much all of the questions, and so I'm going to draw the session to a close at that point and thank all of our speakers for joining us today. Up after the break, we have 3 sessions to choose from: CAR T cells in the clinic on the research track and on the supportive care stream, how neuroblastoma has shaped but not defined me. And with that, we'll move to a break. Thank you all for watching.